Genomics and Medicine, Chapter 4, Principles of Genetic Variation. Module 5, Genetic Variation and the Immune System. This slide depicts the extreme variation that is observed in 4 classes of immunoproteins. The genes that are involved in identifying microbial pathogens are subject to constant positive selection to maximize diversity in the proteins involved in antigen recognition. This involves 4 types of proteins that belong to 2 broad classes, as shown in this slide. Proteins include the immunoglobulins, which are expressed on the surface of B cells in the bone marrow or secreted as soluble immunoglobulins by activated B cells. Their task is to recognize and bind specific antigens. The second type of molecule are the T cell receptor proteins. These are displayed on the surfaces of T cells and work in cell-mediated immunity, along with proteins encoded by the major histocompatibility complex, or the MHC complex. And then there are the classical class 1 MHC and classical class 2 MHC proteins. In humans, these are known as the HLA proteins, and these proteins are expressed on the surface of almost all nucleated cells and enable cytotoxic T lymphocytes to recognize and kill host cells that are infected by viruses or other intracellular pathogens. And you can see in this slide where the variation occurs. So the yellow colored boxes depict the region of the particular protein that is highly variable. The orange dots show you where the polypeptide chains are joined in the large IgG molecule that join the 2 chains together. And the slide below shows how many different haploid genes are there for each particular type of immunoprotein, and gives you an approximate number. And you can see that these numbers are quite large. Each individual can produce millions of different types of antibodies and T cell receptors, and the sources of the genetic variation are also listed and include things such as somatic recombination and hypermutation, as well as just simply extensive polymorphism that is present in the population. This slide summarizes an overview of the origins of genetic variation found in the human immune system. The primary source of diversity is the random and targeted somatic variation that gives rise to combinatorial diversity via somatic recombination. Each IgG and T cell receptor is made up of a series of repeated gene segments that specify discrete segments of the protein, and different combinations of gene segments are used in protein production in different B cells or in different T cells of each individual. The different combinations of gene segments are made possible through somatic recombinations that occur in IgG genes in mature B cells and in T cell receptor genes in mature T cells. Additional variation is found through junctional diversity, the protein chain diversity, and somatic hypermutation. Junctional diversity refers to somatic recombination mechanisms that bring together different gene segments in IgG or T cell receptor genes, variably add or subtract nucleotides at the junctions of selected gene segments. Protein chain combinatorial diversity describes the IgGs and T cell receptors that are heterodimers, and diversity is compounded by unique combinations of protein chains. Somatic hypermutation. This mechanism applies only to IGs and is used to further increase variability in the variable region after somatic recombinations have produced functional VDJ or VJ units. When B cells are stimulated by a foreign antigen, an activated induced cytidine deaminase is produced by the activated B cell that deaminates cytidine to uracil. The uridines are variably repaired by basic scission repair, and the end result is that multiple nucleotides in the variable region are mutated. This slide depicts somatic recombination in the IgH gene of B cells that is used to make specific immunoglobulin heavy chains. The human IgH gene has multiple but slightly different repeats for each of the 4 types of gene segments: V for the first part of the variable region, D the diversity region, J the joining region, and C the constant region. An immunoglobulin heavy chain is made by bringing together coding sequences from each one of these 4 types of segments, shown here as filled boxes. 2 sequential somatic recombinations produce first DJ joining, then a mature functional VDJ coding sequence unit, which is effectively a large novel exon. Another type of somatic recombination, known as class switching, but not shown here, can change the position of the C or constant segments so that other C gene segments can be used instead of C mu to give an alternative class of immunoglobulins. The MHC or HLA proteins. The HLA complex is the human major histocompatibility complex, or MHC. The latter name came from the observation that certain MHC genes are the primary determinants in transplant rejection. That is, in an artificial situation, the normal function of MHC genes is to assist certain immune system cells, notably helping T cells to identify host cells that harbor an intracellular pathogen, such as a virus. Our most polymorphic proteins are produced by genes in the HLA complex. HLA proteins recognize and bind peptides from processed foreign antigens and present them on the surfaces of cells so that they can be recognized by specific T cell receptors. Some MHC genes, called classical MHC genes, are extremely polymorphic. They are subject to positive selection to maximize genetic variation, since people who have heterozygous multiple MHC loci will be better protected against microbial pathogens and have higher reproductive success. This table shows statistics for the 6 most polymorphic HLA loci. Data were derived from the European Bioinformatics Institute's IMGT/HLA database. You can see that a wide number of alleles or DNA variants give rise to an increasingly large number of protein variants in each case. Importance of the HLA system. Certainly, it's true that the wide genetic variation of our immune system confers us with increased resistance towards infectious disease. However, it is not without consequence in medical scenarios such as transplantation and autoimmune disease. After organ and cell transplantation, a recipient's immune system will often mount an immune response against the transplanted donor cells, known as the graft, which carry different HLA antigens from those of the host cells. This immune reaction can be sufficient enough to cause rejection of the transplant. Bone marrow transplants and certain stem cell transplants can also result in something that's known as graft versus host disease, when the graft contains competent T cells that then attack the recipient's cells. Graft versus host disease can even occur when a donor and recipient are HLA identical because of differences in minor non-HLA histocompatibility antigens. Immunosuppressive drugs are used to suppress immune responses after transplantation, but transplant success depends largely on the degree of HLA matching between the cells of the donor and the recipient. Histocompatibility testing, also called tissue typing, involves assaying HLA alleles in donor tissues so that the best match can be found for prospective recipients. The key HLA loci are the most polymorphic ones: HLA-A, HLA-B, HLA-C, DRB1, DQB1, and DPB1. HLA and disease associations. In autoimmune disease, the normal ability to discriminate self-antigens from foreign antigens breaks down, and autoreactive T cells launch attacks against certain types of host cells. Certain HLA antigens are very strongly associated with individual diseases, such as type 1 diabetes and rheumatoid arthritis. In general, genetic variants in the HLA complex are the most significant genetic risk factors that determine susceptibility to autoimmune disease. Determining to what extent HLA variants are directly involved in the pathogenesis and how much is contributed by other variants that lie in the immediate vicinity of the HLA genes is a major area of research. HLA gene nomenclature and haplotypes. The genes in the HLA complex are highly clustered and are confined to an area that represents only about 2% of chromosome number 6. Genes that are close to each other on a chromosome are usually inherited together because there's only a small chance that they will be separated during recombination that occurs in short intervals separating the genes. Such genes are said to be tightly linked, so the HLA genes are tightly linked on chromosome 6. A haplotype is a series of alleles at linked loci on an individual chromosome. The haplotypes were first used widely in human genetics with reference to the HLA complex. This table shows the nomenclature and meaning for the HLA alleles. Haplotypes can be deduced from examining family studies. Father, mother, and their 3 daughters, Zoe, Julie, and Anna, and 2 sons, Bob and Jack, have been tissue typed using serological reagents for 4 HLA antigens, as is shown at the left. By tracking which parental alleles have been passed on to individual children, it is possible to deduce the parental HLA haplotypes. Father has 1 chromosome 6 with the HLA haplotype DR8, B7, CW7, A19, and another chromosome 6 with the HLA haplotype DR6, B27, CW1, A28. Similarly, mother has haplotypes DR2, BB, CW1, A3, and DR4, B14, CW2, and IA9. Father has transmitted haplotype to Zoe and Jack, and haplotype B to Bob, Julie, and Anne. Mother has transmitted haplotype C to Zoe, Bob, and Anne, and haplotype D to Jack and Julie.